Good afternoon. At the onset, thanks to Rajiv for inviting me to this important event. And also, this is for me, this is the lifetime opportunity to be the last speaker just before the concluding session. I try to make the things interesting by not talking on the gene sequencing and all those things that people have been talking. And so the topic for my talk today is drought granule QTLs in rice. At IRRI, we do have a very systematic QTL identification program which is based on grain yield per se. And in fact, this was started well before I joined IRRI. I joined IRRI in 2007 and this was started by Gary Etlin sometime in 2002. But I did have opportunity to work with Gary from 2004 to 2006. And then when I joined IRRI, I did see that probably in rice this is the approach that we should take for a few years to bring some improvement in grain yield. And so we continued this QTL identification program at a very systematic way and probably with the same efforts that Gary had put in earlier. Now this was just not a QTL identification program, but it was a QTL identification program in the background of high yielding varieties. So it was more or less that you identify the QTLs that you can use in the breeding program. And in fact today I can say that we are using in the breeding program 90% of the QTLs that we have identified over years. We also took efforts to validate the effect of some of the identified QTLs in different environments and different genetic backgrounds, with the idea that probably in years from now, when this program was initiated, we have some QTLs which shows effect against multiple background and in different ecosystem and in different environments. Up to now, in fact it's 2014 now, we have identified 14 QTLs. We have reported these QTLs through 17 publications. And in fact now we have started using some of these QTLs in the breeding program. I directly go on the QTL identified that we have identified so far, and this is I start with the QTL 1.1. This QTL is located somewhere near ST1 locus in the rice where the tall allele of the ST1 exists. But what I want to show here is that this QTL we have shown that is coming from at least 4 donors and is showing effect against 3 prominent mega varieties, which is Swarna, IR64 and MTU 1010. And again this QTL is showing effect in both lowland and upland ecosystems. I report not all those 14 QTLs today, but I do report QTLs, 8 QTLs which have shown effect across 2 or more genetic backgrounds and across both lowland and upland ecosystem. And so the second QTL is QTL 2.2, which is again we have shown is coming from at least 2 donors and showing effect against 2 backgrounds and is again showing effect in both lowland and upland. Same as the QTL 2.3, then there is QTL 3.1, then there is a QTL 3.2, then there is a QTL 6.1, and then there is a QTL 6.2, and then the QTL 12.1. In fact, we can go reverse wise. 12.1 was the first QTL that we identified, and then that was subsequently the other QTLs came. But what I want to say here is that you see the genetic background, the recipient background. We never had any recipient background which is not a cultivated variety. So the efforts were not to identify the large effect QTLs, but efforts were to identify QTLs which can be directly used in the breeding program. Knowing something more about the QTLs, this happened by chance. We were screening a population over 2 years, and eventually one season we got very good drought, severe drought, and one season we got moderate drought, and of course we screen all our population under irrigated control situation also. So under irrigated, and this is a story about the two QTLs 2.2 and 4.1. In irrigated control, we didn't see any effect of any of the QTLs. Under moderate stress, we do see effect of one QTL, which is 4.1, and under severe stress, we see the effect of the other QTL, but the effect of 4.1 gone. You call it G by E, you call it QTL characterization, or you call it that we don't know much about all these QTLs effect. So this is what we learned. Later on, we introgressed these two QTLs, and you will see that story in IR64, and we are seeing the effect, improved effect under drought of these of the IR64 drought tolerant lines under both moderate and severe stress. Another good story, good learning exercise for us was that when we identified QTL 12.1, this QTL came from a susceptible background. And the whole idea when Gary made this cross Bandhana Vereram was that we get a QTL from Bandhana, which is a drought tolerant variety, and we will be able to improve Vereram. It happened other way. The QTL we identified was coming from the susceptible variety. But this QTL at that point of time showed effect only in upland. We didn't see any effect in lowland. When we did the epistatic analysis, what we saw was that this QTL was showing interaction with the QTL on 2.3, and when we did the class analysis, we could see the increased effect of those lines where both these QTLs were present over the lines where only 12.1 was present. To our surprise, when we grow these lines in lowland, we see similar results, and in fact we could see an effect of this QTL in lowland also when both these QTLs were present. Subsequently, we identified 12.1 showing effect in lowland also in a different background, which is Savitri, which is a popular variety from Nepal, and it was a different donor, but donor was, again, Vereram was involved in that cross. So there was something happening with 12.1 coming from Vereram and 12.1 coming from the derivative of the Vereram. The molecular biologist Ajay Kohli, he worked on QTL 12.1 gene complex, and he says that there are 10 genes involved which is showing effect, which are responsible for the effect of the QTL 12.1, and in fact the most important among them is the transcription factor OS9. And in fact, when we try to do the fine mapping of this QTL, we have seen that effect of this QTL have reduced, whether we have used the SSR-based marker or we have used the candidate gene-based fine mapping. We have seen that we are getting a different fragment. And then this led me to think, what's happening with those candidate genes where people are talking of the effect of one gene? If this is the effect of 10 genes that we are seeing in a QTL, which have shown effect of somewhere around 500 kg, is individual gene responsible for 50 kg, or do we still have genes where single gene can show effect of 500 kg? We got some more surprises. When as I grew these transgenic lines, he did see that the transgenes with the Vereram OS9 have shown similar phenotype as that of the Niels, where we have introgressed the QTL 12.1. However, he was not able to see the same yield advantage that we saw when we introgressed the QTLs in the Nil. And then probably this was because of the interaction between QTL 12.1 and QTL 2.3, which was not there. The second thing that we learned from here is that we did see the effect of the QTL was because of the increased number of nodal roots and increased spikelet fertility. Now surprisingly, this increased number of lateral roots and root hairs was happening only under drought. So if you have normal situation, you don't have production of those increased number of lateral roots. And so again, the second surprise for us was that is that the mechanism that helps us to identify lines or develop lines which combines good yield as well as better yield under drought, or we should just look for the root the root structure where we have more number of lateral roots, or this is just specific to QTL 12.1. Moving further, when I started working on drought in 2004, I thought that probably not much effort had been paid to drought breeding, and I will bring a significant change in rice and drought. But when I see it back after 7 or 8 years, I do see that people have made tremendous efforts earlier, and there were bottlenecks which people were not able to handle earlier. But now we do have technology which allow us to handle some of these bottlenecks. So I still keep this open, whether these are facts or assumptions, but we do know that most of the drought-tolerant germ plants are tall in height. Whether there is a linkage between drought tolerance and tall height. Most of the drought-tolerant germ plants, they possess lower yield potential. Is there a linkage between the two? Most of the drought-tolerant germ plants are early in duration. Means I do see that when we started this drought breeding program, there is a shifting towards the earliness of the whole breeding lines that is coming from the drought-tolerant breeding program. Some people also say drought-tolerant genotypes are photosensitive, and drought-tolerant germ plants have efficient root system. I leave the last two, but I do talk about these three in detail, what we see in our QTLs. Effect of QDTY QTLs on an undesirable trait. I said earlier that QTL 1.1 is located somewhere near ST1, tall allele of the ST1, and we do see effect of this QTL on tall plant height as well as earliness. We have seen effect of QTL 3.2 on early maturity. We have seen effect of QTL 3.1 on reduced yield under irrigated system. We have seen the effect of 6.2 on again tall plant height. The question is whether this is pleiotropy or linkage. If this is pleiotropy, forget this. We will continue to have this problem. Or if this is a linkage, then can we break this linkage and develop drought-tolerant varieties which are high yielding, which are not tall, and which are not so early in maturity. We developed mapping population for QTL 1.1, where we kept the ST1 allele fixed, dwarf allele, so it was a dwarf population, and we were able to fine map this QTL somewhere below ST1. I am not putting the names of the markers as of now because we are still finalizing this, but we know from that backcross inbred population. We also put markers on our real population, additional markers where we earlier identified this QTL, and we now know that QTL 1.1 is well below the ST1 locus, and in fact we have broken this linkage. We have also shown that QTL 1.1 effect in the dwarf population is similar to what was the effect in the tall population where we initially identified this QTL. We did see similar effect with other QTLs. In 3.1, we do see decrease in yield under normal situation. 6.2, we do see increase in height under both normal as well as drought situation. With 3.2, we do see drastic decrease in days to flowering under normal situation. For 3.1, for 1.1, we did break it by developing that bill population, but for other QTLs, we went back to the original mapping population and we did class analysis. Now you can see here that for QTL 1.1, dwarf plants which were similar height as that of Sorna under non-stress provided similar yield advantage over Sorna that we have seen earlier, and with no yield reduction under the normal situation. Same thing for QTL 6.2, which we identified in TDK background. Lines with the similar height as that of TDK showed effect under drought with no yield penalty under the normal situation. For 3.1, we were able to break again that linkage with undesirable reduced yield, and you can see that lines, we were able to develop lines which has the similar yield potential as that of Sorna and provided yield advantage over Swarna. For 3.2, our efforts were to identify lines which had higher days to flowering than that of Bandhana, but still we see the increased effect under yield over Bandhana. And this we were able to show. So we have data for these 4 QTLs, that these QTLs are linked with some of these undesirable traits. And we can break these, good thing is that we can break these linkages, and we have opportunity to transfer these alleles, the fine mapped alleles into the dwarf backgrounds, and we can develop dwarf plant type with the increased drought tolerance. We did one diversity analysis with the 11 QTLs, 55 alleles, and it was 130 lines, which included drought tolerant donors, which included the green revolution varieties, which included the recently developed drought tolerant varieties. I thought that, and we used 55 markers for the 11 QTLs. I thought that probably we will get maximum 2 or 3 groups, but what we got was 5 groups. Group 1 was, of course, the drought tolerant donors, which includes N22, Adacel, and all those donors that we have used. Group 2 were the early generation green revolution varieties, IR8 and derivatives. Group 3 was slightly, I think, similar varieties, but it separated from IR8. I don't know for what, DGO and Jaya, which were again the early green revolution varieties. Group 4 are the mostly varieties which is being cultivated now, including IR64, MTU1010, Swarna, and group 5 was the drought tolerant varieties which had been developed recently. So this surprised me, and then we decided to see that what is the allele frequency in all these different groups of the drought tolerant alleles as well as the recipient alleles. And to my surprise, what we see is that there has been a continuous reduction in drought tolerant alleles as we have moved across the green revolution era. This has reduced from 39% to 15%. We have lost these drought tolerant alleles as we have bred for. On the other side, we have the recipient percentage of recipient alleles have gone on increasing. And this is what we have done for the rain-fed ecosystem when we have bred for the irrigated ecosystem. Our efforts at IRRI, we were able to bring back some of those drought tolerant alleles. But this is the story with the major alleles. What happened to all those minor alleles which we are not able to detect now, and what happened to all those interactions where they are lost, and how do we bring them? Are similar things happening in other crops? Earlier we have reported that QTL 1.1 region in rice is sentinel to QTL maize chromosome 3, wheat chromosome 4b, and barley 6H, and QTL 3.1 region is sentinel to maize 1. And there are QTLs reported in these regions in different crops for the drought tolerance as well as the traits, undesirable traits which we discussed, some of the undesirable traits. But I don't have a story to say whether this is same thing is happening in wheat, maize, and barley. But we do have a story to show that I showed you that what is happening in rice. Moving ahead with the use of the QTLs in the breeding program, we initiated a massive QTL integration program. Our efforts initially were that we use QTLs which we have identified so that we are better sure of that we do get an effect of these QTLs. But as we became more confident, we started using these QTLs across any background. And today I show you quickly the data from the 5 such integrations that we have. And these integrations are at a different level. Bandhana, this is being evaluated in the state varietal trial in Jharkhand. IR64 QTLs already completed the active evolution for 2 years in India. And in fact, it has already been evaluated in Nepal. And at the verge of release, it has been identified for release, and probably it will also be released in Jharkhand very soon. And it's being tested in other countries. And other QTLs are at the different levels. So with Bandhana, those of you who have worked on rice very well know that bandhana is a drought tolerant variety, and there could be no better case to validate the effect of drought QTLs if you can improve the yield of a already drought tolerant variety. This QTL was introgressed at IRRI in bandhana. At IRRI, we do see effect of 0.5 ton increased yield over bandhana. We tested, we sent these lines, we tested these lines across India at several locations, and we do see effect based on the severity of the drought. So, and in fact it has been reported earlier that the effect of this QTL increases with the increased severity of the drought, and we are seeing a similar effect for the QTL 12.1. IR64 drought, where we have introduced the two QTLs, 2.1, 2.2 and 4.1, is a similar story. We are seeing, in fact, these QTLs, these lines have been screened at 51 locations across India, Nepal and Bangladesh, and we did see yield gains over IR64 at 46 of those locations. At those locations where we had severe to moderate drought, yield increase was ranged from 50% over IR64 to 450% over IR64. It is very interesting in this case. We started with, in fact, 84 lines which had similar yield advantage, and this was a ABQTL analysis approach. And then we analyzed those lines for 2 years. We came back to 10 lines, and then we did the cooking quality analysis because we didn't want to lose on IR64 cooking quality, because IR64 is popular among people because of its cooking traits. And we could find only 3 lines which had the similar cooking quality traits as that of IR64. And so we moved ahead with testing those 3 lines. Then, getting confidence, we started improving Swarna. In between, Swarna sub 1 was released, and it was getting very popular in different places in India, Bangladesh, Nepal, and we switched our approach to improve Swarna sub 1. And by that time, we also had QTL 1.1, and so we introduced 3 QTLs in Swarna sub 1. And we do see similar effect what we predicted earlier, and these are the 3 lines which now we are targeting to have both sub 1 as well as the drought tolerant yield advantage. And there are other lines which do have yield advantage, they don't have the sub 1. These were the 3 examples where we introgressed the QTLs in the background, where we did see a effect, where we identified the QTLs. But then we started testing these QTLs across other backgrounds. We introgressed the 2 QTLs where we see the effect in IR64 into Samba Masuri, and we do see a similar effect in Samba Masuri, what we have seen in IR64. We took 2 varieties from Malaysia. It was a scholar from Malaysia, and she introgressed 3 QTLs in these. And again, these were the new background, and we do see similar effect over in these 2 backgrounds also over the recipient varieties, yield advantage under drought. We have never compromised with the yield potential under normal situation. So now we suggest that probably these QTLs can be used across any genetic background, but with a caution that they may not be the best QTL for that background. So you can identify the best QTL or QTL combination for that background, but what I can say is that definitely we are going to get some yield advantage by using these QTLs across any background. But we have to be very careful in devising the breeding program. We know less, we don't know more. Still, I believe in that theory. We don't know QTL into QTL integration. We don't know QTL into genetic background integration. How we overcome that? By developing a large BC2 or BC3 F2 population, where we allow breeder to capture that positive interaction, and you screen a larger BC and F3 population under drought. So many of the undesirable linkages which you don't know, you can break here and select for plant type, and you can capture the positive interaction between different QTLs and QTL and genetic background at this level, so that then you make appropriate breeding modifications if you have a larger population at this place, and then associate that with the genotype and phenotype through standard phenotyping, probably 2 seasons of phenotyping. You don't get everything in the first season. This is my last slide, and I'm opening probably a controversy, making a controversial statement. Is rice different from other cereals? We haven't seen too many large effect QTLs in wheat, maize, barley reported, and we haven't, at least I have not seen any report where we say that we increased the yield by 1 ton under drought. To me, it means that there has been clear-cut examples. We introgress 1 QTL in Bandana and we see 0.5 ton yield effect. And I say again that you are seeing this yield increase over a Bandana, which is a drought-tolerant variety. There is no program which, as of now, which addresses that we are improving a drought-tolerant variety. So there are many programs which say that we are improving a drought-susceptible variety, and probably we would not have done it. This came as a surprise to us. But then we took that challenge and we say, let us see that if QTL is real, can it improve that drought-tolerant variety? We put 2 QTLs in IR64 and Samba Masuri, and we see 1 ton to 1.5 ton yield advantage. We put 3 QTLs in Swarna, and we see 1.5 to 2 ton yield advantage. Now, we may have been, this may have been by chance that we have put in the best combination of QTLs, but we still don't know that what are the best combination of QTLs for different genetic background. What happened, what I have explanation is that probably rice is differently placed than other cereals. Rice has now still been grown under 2 situations, upland and lowland. We did an exercise earlier where we screened a number of upland lines, and we could see that many of our identified QTLs are present in high frequency in upland lines. And this is assumption that probably these drought-tolerant alleles are still existing in some way and are under the upland varieties which are grown, still under some sort of drought situation, and they carry these drought-tolerant alleles. And in fact, if you see this whole QTL breeding program, most of these QTLs are coming from those upland adapted germplasm. And on the other side, we have grown rice under transplanted situation with ample amount of water, lost those drought-tolerant alleles, and have increased the susceptibility of the post-Green Revolution varieties to drought. And thereby, we have a duty to bring back these drought-tolerant alleles to the lowland varieties, which probably this kind of differentiations don't exist in wheat or maize, where they are grown in more or less similar situation. Maybe I'm wrong, but this is what we see. Why do we see such a large effect under rice, but we don't see such large effect in other cereals? With that, it's time for me to say thank to all people who worked with me at IRRI over last 7 years, including the molecular biologist and physiologist, and then people who worked with Gary, including myself, for 2 years, and people who worked with Gary before I joined the rice breeding group, rain-fed lowland situation at IRRI. Thanks to all the partners across South Asia and also some in Africa, with whom we have worked, and we are testing these lines. And I can make a statement here that this continuous work over last 12 or 13 years would not have been possible without the continuous support from the donors. Whom I name first, Generation Challenge Program or Bill and Melinda Gates Foundation, it's a challenge. Both of them have supported us tremendously, continuously over years, from 2004 to 2014. And in fact, we say the GCP is going to have a sunset. Bill and Melinda Gates Foundation continue to support us for the next 5 years. That is what they have promised. We did get some support from DVT, Devjan, which has become Syngenta. We had support from RDA Korea and BMJ. Thank you very much for your attention. Thank you.
Fortunately, because we're running late and the next session does have to start probably at 3:30, we have no time for questions. So if anybody does have questions, maybe they could ask them during the coffee break.
Why this unfavor to me? Just a joke.
Just for you, yes. And I think Anu has a announcement